Welcome to those of you who have joined us in the last few minutes, and welcome to everybody to this Proteoform Thursdays by the Consortium for Top-Down Proteomics. I'm Ollie Hale. I'm a member of the Early Career Committee for the Consortium for Top-Down Proteomics and a postdoc at the University of Birmingham in the UK. It's a pleasure to have Pei Su with us today from UC Riverside, and he's going to be presenting a talk on spatial top-down proteomics. So I think this will be very interesting. First of all, I'd like to thank our sponsors for supporting the Consortium for Top-Down Proteomics. We have a number of events coming up, including a workshop in early February, which is a couple of hours, and then a talk in March as well, which is a top-down. It's a Proteoform Thursdays event. We also have the ECR committee rolling over to its third term at ASMS this year, and nominations are open for that. We have the form for applications and nominations on our social channels, so if you look for those, say on LinkedIn or Blue Sky, you'll be able to nominate. You can self-nominate for that if you're interested. So this is the workshop that's at the start of February. So it's looking at top-down within and extended, so native as well, within the biopharmaceutical characterization space. So yes, it's a pleasure to have Pei Su with us today. He attained his BSc in chemistry in Shanghai before moving to Purdue for a PhD in analytical chemistry with Julia Laskin. He then moved to work as a postdoc with Neil Kelleher. I'm sure everybody knows Neil in the top-down space at Northwestern University, and now has begun his independent career at UC Riverside. Pei, over to you.
All right. Thank you so much for the kind introduction, Oliver, and thank you so much for this great opportunity to present and talk about my work. I just want to make sure that we can see the presenter mode.
Yep,
Yes.
that's
Does
great.
everyone see my screen okay?
Yeah, we can see it.
Okay, awesome. All right. Good morning from West Coast of the United States. And as Oliver just introduced, I started last November as assistant professor at UC Riverside. Today's presentation is mostly still about my work conducted as a postdoc in Neil Kelleher's group at Northwestern University. Our focus was from using single molecule mass spectrometry approach, high sensitivity detection, applying that to spatial and single cell proteomics. Apologies in advance. I got flu second time in a row in this month. I guess I haven't gained West Coast immunity. If the voice doesn't go well, and I'll try. All right. One of the ultimate goal in biochemistry is to map the molecular avatar of biological systems, and that includes the mapping of a multitude of networks from organs to cells to molecules. To really describe this kind of molecular avatar, we really need to develop spatial and single cell molecular omics technologies that allow us to quantitatively map molecules across different anatomical regions, different cells across the space. And this includes the detection of different classes of molecules as well, from nucleic acids to proteins to lipids, carbohydrates. Sometimes we categorize them as metabolites. And my focus during postdoc is mostly in the protein space. As we all know in this audience, that proteins are not just the amino acid, not just the combination of amino acids, right? So they are proteoforms in human systems that from the 20,000 genes, we going through the mRNA transcription process, we make proteins, and then these proteins are exposed in a very complex and dynamic biological environment that will be modified post-translation, right? So this includes, side chain modifications such as glycosylation, phosphorylation, and then this could also include other types of modifications that make a myriad of different molecular forms of proteins. And we use proteoform as the term to describe this complexity of molecular landscape. And so proteoforms have been recognized as important disease markers. And in our consortium, we have made a lot of amazing progresses in connecting proteoform signatures to disease phenotypes. So a few examples include the ones that are listed here from neurodegenerative diseases, tau proteins that are hyperphosphorylated, that are directly connected to Alzheimer's disease. And we also have like cardiovascular disease that phosphorylated troponin reports the cardiac injury, and a few other examples. So, from a fundamental kind of a biochemistry perspective, I would like to actually motivate this audience from a slightly different angle that we can really use proteoforms to inspire a discovery in biology post-translation, right? So not from the gene and mRNA level, but actually from the protein level post-translation. So the one example that's from Neil's lab is the triphosphate isomerase (TPI), and this protein we all know that it's the racing car enzyme in the glycolysis process, very high catalytic rate. And but then if you put a phosphate group on the number 20 serine of TPI, then you can still make the catalytic activity like faster by 4 to 5 fold. Right. So, and this means that, adding a phosphate group to this TPI protein molecule is not just adding a chemical moiety, but it also induces significant structural changes to this molecule that can have important biological implications. Another example is also from our community. Professor Julia Chamot-Rook. These, bacteria that can infect the brain, they express a protein called PoE on their surface. And this protein can, out of an ocean of different, small molecules, it can hijack this glycerophosphate to make a glycerophosphorylated form of PoE, and then eventually allow this bacteria to cross the blood-brain barrier and infect the brain to cause this cerebrospinal meningitis. So this is just an example of like how a detecting a whole protein, understanding its modification type will allow us to connect very complex disease phenotypes that could be happening with a variety of different biological environment. So here actually talk about proteoforms that are important disease signatures. But then, like, I guess you might have a question of like, why do we want to do spatial detection or detection in specific like contexts such as single cells, right? And this is one example of in my postdoc study that we actually work with like a project with ovarian cancers. And ovarian cancer is among the highest mortality type of cancer. This still does not have a effective marker to diagnose. And so we got this section of ovarian tumor that is a very has a very spatial complex spatial context, including tumor region outlined by the by red, and the stromal region outlined by blue, and also some vasculature structures in the middle. And so from our spatial proteoform platform, we were able to detect this cysteine-rich protein 1, CRIP1, that has three different proteoforms that we can see in the spectrum here. So each highlighted cluster of isotopes, they correspond to one proteoform of CRIP1. And by doing database search and intact mass tag annotation, we learned that this first proteoform is the canonical one that does not have any modifications. And then the second one in the middle has a single methylation that's monomethylated, and the one on the right is doubly methylated. So when you look at the spatial distribution of these three proteoforms, you will see these very drastically different distribution in this tumor section. That the unmodified CRIP1 is localized to the vasculature region highlighted here, but then the two other modified forms are more evenly distributed. So this actually indicates that we really need to resolve molecularly the three proteoforms to be able to like tease out these different spatial distributions, and then you can make use of this modified CRIP1 as a marker for the vasculature, which indicates the tumor angiogenesis and tumor growth. And moreover, you can see this as like a activity-based spatial profiling, which means that the uh, the CRIP1 proteoform that are methylated, they are not the method groups are not added by random processes, right? So they are modified by specific enzymes that are called methyltransferases. And so this means that this different spatial distribution indicates the different activity of methyltransferase across different tumor regions. And the activity of this methyltransferase is actually suppressed in the location of the tumor growth. So this can basically give us a handle of drug development that you can think about methyltransferases as a way of like a target that you can modify and to boost its activity to suppress the tumor development. So a spatial single cell proteoform detection is amazing, but it hasn't been done like extensively, and this is because it has been a challenge. So if you look at this bar graph of copy numbers of mRAs and proteins in a average single cell, so mRA has an average of 17 copies. It's not many, but we have polymerase chain reactions that can amplify and achieve effective detection of these molecules. But proteins has a median of, 50,000 copies, but we still don't have a effective amplification process to really detect them. So we still have to work with the detection of these molecules directly, right? And then also the distribution of molecular copies of proteins is also really wide compared to mRAs. So this means that we have to be able to detect these low copy numbers in the presence of these extremely high copy numbers of proteins. The wide dynamic range also poses a significant challenge for protein detection. Moreover, we talk about this, 20,000 genes, transcripts, and eventually to proteins and modified to give all the proteoforms. So this, it has been estimated that in a single cell type, we could have more than 1 million different types of proteoforms, just because of this complex biochemical environment and possible modifications on the proteins. So this is challenging, but we have been working on it in the community for a long time. So top-down mass spectrometry is the way to look at proteoforms as intact species, so you can describe them as a as a whole molecule, and then you can dissect that into the sequences and the modifications and the location of these modifications on specific residues. So the typical workflow starts from like cells and tissues. We perform extraction of proteins without perturbing its chemical content to achieve intact proteoform extraction. And then the typical workflow is to convert them to ions via electrospray ionization, and typically with the assistance of liquid chromatography for separation and enrichment, also including capillary electrophoresis. And then eventually we send these ions into mass spec for the molecular mass determination of the intact species, and then gas phase fragmentation of selected species to achieve sequencing of the protein IDs and the modification sites. And this is typically done by bioinformatics approaches with databases. So top-down mass spectrometry has this ability to directly report molecular mass and the connectivity of sequence and their chemical moieties, modifications. It has this quantitative to semi-quantitative power. You can detect thousands of proteoforms in one experiment. It's high feature capacity, and also the throughput is also relatively high with the, you can, if you run a fast LC gradient, you can achieve like one sample like in every hour or even less. So the one thing in doing top-down mass spectrometry using electrospray ionization mass spectrometry is that we typically perform this kind of mass spec detection in the ensemble mode, which means that when we perform electrospray ionization of a protein, it's going to generate a distribution of charge state of the single mass. And the way that we calculate the molecular mass from a single protein species is by looking at the adjacent charge states. And then you can derive the mass using the equation shown on the left. So this is an example of a single like a species that's bovine ubiquitin that has these charge states from 13 plus to 5 plus. And the way to calculate the molecular mass is to by looking at these selected two adjacent charge states, and then you calculate the mass constraints by one different charge state, and then you can derive the molecular mass. So this is effective when you have a simple spectrum, but then when you start to look into complex mixtures, the spectrum becomes very quickly like complicated and hard to resolve. And moreover, this relies on the detection of the same protein in the same scan with many, many copies that generates a constructive interference and give you these different charge states that you really have to have all these charge state neighbors to be able to see the adjacent mass constraints and determine the molecular mass. So, with the advancement of Fourier transform mass spec, including the Fourier transform ion cyclotron resonance and the Orbitrap mass spectrometry, so this allows to achieve high resolution detection of, intact proteoforms in a single charge state. So without looking at just one peak of a charge state, you basically split that into isotopologues if you zoom into one charge state of that ubiquitin shown on the right. And these isotopologues can help you determine directly what the charge state is for this particular mass. And then from here, you can calculate the molecular mass of this protein. So this is amazing, and this made a significant change from the previous type of detection. But then we still have to have thousands of copies of the same ubiquitin molecule running in, for example, the Orbitrap chamber at the same time to generate this type of constructive interference, and then eventually give you the isotopologues for the mass determination. So In the case of Orbitrap-MS, actually this analyzer has the ability to detect single ions instead of thousands of ions at the same time. So this is an example of a protein called pyruvate kinase. It's 232 kilodalton. It's a tetrameric complex. And when you do native electrospray ionization, you will see these five charge states from 36 to 32. And then when you dilute the sample and drop the concentration and introduce less ions into the Orbitrap at the same time, you will see these the transition from these peaks, single peak of a charge state, into these discrete bars that are aligned to these charge states. So these are actually all single ion detection events from the pyruvate kinase single tetrameric molecular ions. Um, and you might also notice that the height or the intensity of these bars, they have a proportional relationship that basically higher charge state is able to induce more signal, like higher signal intensity for compared to the lower charge states. So this and here I'm just showing a trend to guide the eye for the intensity proportional relationship that I was talking about. So while I was in Neil's lab in the past few years, I had this great opportunity to participate this collaboration between Thermo Fisher Scientific and to really push forward the single molecule detection of intact proteins in Orbitrap systems. And we developed this technology called individual ion mass spectrometry. The acronym is I2MS. And the commercial format of it is direct mass technology mode. That's available now on the UHMR system and also the Orbitrap Ascend systems. So basically, the principle is relying on this like a simple math equation where you determine the mass to charge ratio of a single ion. At the same time, you measure or determine the charge state of this ion. And then by multiplying this charge state and the mass to charge, you get the molecular mass of this ion. And the charge state is determined by the induced signal over time as you trap that ion in the Orbitrap. And this gives you the story plot that has a slope that is proportional to the charge state. And this is actually quantitative charge determination that can separate one charge state from the adjacent one. And eventually, by running this kind of equation for every single ion, then you can build a histogram of your protein detection that with all these different like molecular masses, you can build an isotopic distribution from scratch using single ions, and eventually get this isotopic distribution that can be correlated to the intact mass of this proteins or proteoforms. So this is one example of just showing how it works in a typical experiment. And we published this paper on Nature Protocols last year just to like guide an experiment from getting a sample of a like an intact antibody sample, how to run it through native electrospray ionization to the I2MS DMT platform, and then eventually get the complex mass landscape of the proteoforms of the antibody species. So basically, what you do is that you do continuous injection of the antibody sample using native electrospray ionization. And you, in every spectrum you get, every scan you get, you'll get these single bars that are single ions of the intact antibodies, and different groups indicate different charge states. And then you continuously acquire the spectrum of the same sample, and then you will get more and more spectrum representing different discrete events of detection of the molecules of the antibody. And eventually you do individual calculation of all of these mass of every single ion. And then you will generate this histogram masses and eventually build up these isotopic distributions. So this is quite exciting for a molecule that's close to 150 kilodalton. You can still achieve isotopic resolution of the detection, and these different clusters of isotopes can be aligned to different glycosylated forms of the antibody that can be correlated to these like exact forms that so not only tells us that I2MS and DMT mode is amazing in resolving the isotopic clusters of proteoforms, and also it has this ability to detect really high mass proteins and their proteoforms. All right, so I talked a lot about the single molecule approach using Orbitrap-based I2MS. Then it has this amazing sensitivity to detect single ions, and also it achieves a very broad mass range to like very high mass proteins that you can see. But then we kind of haven't really talked about how this connects to the spatial and single cell proteoform detection, right? So and actually, so like conceptually, you would think about like to achieve spatial and single cell proteoform detection, you will actually need to use a spatially resolved probe that ideally have a high spatial resolution to resolve a cell from the other. And also, you want to achieve a high throughput that can run many different pixels within an hour. And also, ideally, we want to achieve a high capacity detection of many proteoforms from a single sample. So basically, you have to develop this highly multiplexed approach to see many proteins in the same experiment. So my major contribution in the postdoc phase in Neil's group was to couple a spatially resolved probe with Orbitrap-based I2MS approach. So the technology we use is called nano-DESI, nanospray desorption electrospray ionization. That's invented by Professor Julia Laskin at Purdue University. So this is a spatially resolved liquid extraction surface sampling probe that is suitable for not just proteins, like many other different classes of molecules. And you can have your solvent of choice to run through the system to extract your molecules of choice from tissue. And The setup is rather simple, that composed of two capillaries that one delivers the solvent to the tissue, and the other one like extracts the molecules from the tissue, delivers the solution of the extraction, and then ionizes at the other end and couple that to mass spec detection. So at the location where the two capillaries meet, you will form a liquid bridge that dynamically extracts the spatially localized molecular content from a tissue. And while you're moving the tissue under the probe, then you can achieve this sequential extraction of different locations with minimal carryover. So the way that we couple nano-DESI to the single molecule approach is to basically still like run this kind of raster line scan across space, and at every single scan here from a location on the tissue, you collect single molecule like I2MS data. So in one spectrum, you get like thousands of ions, and if you zoom in, you will see those individual ions that have different MRZs, different intensities later can be correlated to charge states using story. And then eventually you can just compile all the information from different scans and put together mass histograms. And then these are all spatially encoded, mass histograms. And after you register these isotopic clusters with different proteoforms, you can back project your spatial code to how many of that proteoform ion you detect in a specific pixel on the tissue, and then you can generate this kind of 2D histogram corresponding to a specific proteoform, right? And this process as long as you can detect, hundreds of proteoforms from a single spectrum, you can all just correlate them to the same tissue, generate hundreds of images from a single piece of tissue. And this is just to show you like a gallery of, different proteoform images you can generate from a single tissue section. And the first project that we kind of demonstrate the proteoform imaging mass spectrometry approach, PIMS, that's how we call it, is through this HubMed consortium that we had amazing collaborations with Vanderbilt University and Washington University at St. Louis. So we got those kidney tissues that have basically. So we run these kidney tissue imaging, and we got more than 1,800 proteoforms from a single piece of tissue across four orders of magnitude. And among all these detected proteoforms, 400 of them generated meaningful images that can be correlated to different anatomical section regions and cell types. So in this example, I wanted to highlight on the right side that we actually were able to detect four key enzymes in the glycolysis process um, that and generate images of these four enzymes that shows even distribution across the tissue of cortex regions and medulla regions in the human kidney. And So this work has been highlighted on Chemical Engineering News in 2023 as one of the key applications in single molecule mass measurements using different modalities, including Orbitrap mass spectrometry, and also the spatial proteomics has been a really kind of emerging topic in the community of biology, and it has been highlighted as the method of the year for 2024 at Nature Methods. So, so aside from detecting proteoforms in different anatomical regions, we can also achieve relatively high spatial resolution or moderate to high spatial resolution to be able to resolve proteoforms in specific cell types. So in this case, this is the sample we got from WashU, and we were able to achieve less than 20 micron spatial resolution, approaching single cell level. And in this case, uh, like aside from just resolving proteoforms that are localized to the cortex region here in the middle and into medulla region of the human kidney, we can also resolve some proteoforms that are localized to a specific cell type. For example, in this case, alpha crystalline is localized to loop of Henle that are a specific segment of the nephron in kidney that indicates this amazing spatial resolution that we can achieve. So with all the images that you generated from the single piece of tissue, you can color them differently and then overlay them to generate this multi-channel images that can help you further highlight different anatomical regions and cell types. And in this case, we are comparing the multi-channel PIMS images with the autofluorescence, and that shows a great correlation between these different anatomical regions and cell types. And we were able to contribute our proteoform images from PIMS to the HubMed consortium to build up this domain knowledge of human biomolecular atlas at near to approaching single cell resolution. And as you imagine that, every scan generate thousands of, single ions, and eventually we have to compile all these information from different spatial locations into proteoform images, and the data size is huge. So to achieve a high throughput, not just in the mass spec detection space, but also in the data processing space, we actually had this amazing, bioinformatics infrastructure at Northwestern, and I was able to work with, you know, the team over different stages to really build this kind of informatic powerhouse for PIMS. So routinely we generate 50 million single ions from a single experiment in about 7 hours from one piece of tissue, and then we are able to actually you know generate we are able to convert the spatially localized uh, single ion scans into the proteoform mass spectra, and then pull the 2D heat maps proteoform images for each of these proteoforms, and then eventually compile them into a format that you can use basically on a like different vendors, different visualization softwares, which is like IMZML is the vendor neutral format for the mass spec imaging community. So this process has been like highly efficient. We were able to really effectively convert this into a stack of images to visualize them and find the interesting features. And we also actually built a custom software called Mass Build to visualize the data in-house. And moreover, because we don't have the luxury to do MSMS sequencing for every proteoform we detect from the tissue, we pursued a intact mass tag approach based on the intact proteoform masses to infer the proteoform ID with low false discovery rate, facilitated by a Bayesian approach. And a tailored proteoform database. And moreover, we build a proteoform knowledge base that generated about 500 highly curated kidney proteoforms from multiple experiments from healthy human kidney. And these are the proteoforms that you can use for the future samples that we know they are high frequency kidney proteoforms that we can achieve direct ID for a new sample. So this whole workflow allows to process the data and convert it into something intelligible for different communities in a relatively high throughput manner. And I talked about this challenge of not being able to do MSMS for every proteoforms, but we did try to do on-tissue MSMS to at least identify some proteoforms that we don't know from the proteoform database that we have. So we developed this AutoPIMS platform that with these major two of these amazing computational mass spectrometrists. So we basically run a data-dependent scheme to first generate a survey line scan from the tissue, and then on the adjacent line, we perform DDA MSMS acquisition for a selected list of target proteoforms that we found from the survey scan. And this is actually done through a sophisticated algorithm that determines which spatial location we should be targeting which MRZ window in a multiplex manner. So in one line scan, you can be working with more than 20 targets. So basically, with this integrated platform, we can achieve like at least 70 proteoform IDs from a single piece of tissue with a speed of less than 1 minute per proteoform ID. And this actually goes back to the ovarian cancer project I was talking about earlier, the CRIP1 proteoforms that you might remember. So I talked about this idea of activity-based, spatial profiling, where you can infer the enzymatic activity by using spatial profiling. But then with the AutoPIMS workflow, we were able to further kind of verify and narrow down the list of the activities. So This is the same graph that I showed earlier from CRIP1 proteoforms, 0, 1, and 2 methylations, and the spatial distributions that shows a drastic difference in the vasculature location. And we actually talked about that these methyltransferase activity is suppressed in the location of the vasculature, as you show here. But then with the ability to do MS on tissue, we further verified that the methylation of CRIP1 is actually on the arginine number 68. So this actually allows us to verify that this methyltransferase is highly likely an arginine methyltransferase. And then this allows us to really, if you were to design an experiment to pursue a protein as a target for the suppressing the tumor angiogenesis, then you will have a much easier time to set up your experiment instead of going over a list of dozens of proteins that can be methyltransferases. You can just directly go to the arginine methyltransferases and really simplify your workflow and the experiment time. So we talked about detection, identification of proteoforms in the spatial context. We also wanted to talk about quantitation ability of PIMS. So along the same line of the ovarian cancer project in collaboration with Tom Conrad's group at Innova Hospital. So we actually were interested to see what are the proteoforms that can help us differentiate the tumor and stromal region of the same patient. So in this case, as you might be quite well versed with, quantitative proteomics, so you need many samples from the two different biological contexts, and then generate this kind of differential plot with statistical significance to verify these different proteoform markers, right? And in this case, we are able to use this spatially sampling probe to achieve quantitative sampling of tumor regions and stromal regions, 200 samples of each, and then eventually, generate this volcano plot in the middle that compares the tumor regions and stromal regions and quantified more than 300 proteoform signatures that indicates either tumor enrichment or the stromal enrichment. So I wanted to mention that this is from a single piece of tissue that's like 10 micron thickness. And if you calculate the weight of the tissue, it's actually less than 20 micrograms. So instead of using milligrams of samples in the traditional bulk proteomics workflow, we can significantly reduce the input amount of samples, but still being able to achieve hundreds of proteoform signature quantification. But more than that, I also wanted to kind of pull like one specific example from this volcano plot that the same CRIP1 proteoform that does not have uh, a modification. So this actually shows uh, more tumor enrichment compared to stroma, right? And if you go back to this spatial plot, you might notice that actually because of the dynamic range of the color of the tissue, right? So the color is highly saturated by this vasculature region, but if you remove that, you can still see this enrichment of tumor compared to the stromal region. And this actually is reflected by this violin plot that shows that on average of the 200 samples we got from tumor and stroma, you do see this significance statistically from tumor region that it is enriched, despite being very broad, kind of like a distribution of ion counts from tumor. But and as you might see that it is also because of this complex spatial distribution that's not quite marginalized just by the tumor and stroma regions. So, but nevertheless, by just detecting 10 to 20 copies of this ion of CRIP1, you can already start to kind of tease out this biological difference between the tumor and stromal regions from the single patient. And I hope I was able to inspire this, a single molecule approach for the detection of spatial proteoforms. And we kind of asked the question with a series of experiments from the whole tissue detection in the tissue section that requires about 1 milligrams of total tissue content and about 15,000 ions from the entire tissue for one proteoform. Down to where we do this kind of quantitative proteomics experiment that only uses about 10 micrograms of tissue that has a total of 3,000 ions. And then if you look into that example I was talking about, just the CRIP1 proteoform, it has only about 20 copies of ions in 500 picograms of that 1 pixel, like 1 unit of tumor sample. So this kind of give us the hope to really get down to really test the limit of sensitivity of PIMS and eventually walk into the regime of single cells. And if you look at the amount of proteins from a single cell from previous studies, actually they are in hundreds of picogram regime. So we thought maybe we should give it a try to our technique and see what we get from a single cell. And as you might know, that this booming field of single cell proteomics that has been used that has been matured over the past a few years with bottom-up proteomics workflows. So basically, you start with single cell preparation that you dispense isolated single cells into discrete microchambers in like compartments on a sample substrate, and then you perform chemical treatment, including digestion, alkylation, and all that to get the peptides prepared for the chromatographic separation. Eventually, run it through electrospray ionization mass spec detection, and then data analysis and informatics to correlate your peptide detection to proteins. So this is an amazing workflow that has been done, matured over like uh, different labs and different parts of the world. I've seen how much great significance that has been achieved. But so we thought that maybe using the PIMS approach, it has a unique advantage to bypass some of these sample preparation steps that can directly take us from the single cell isolation stage to proteoform detection and eventually correlate the proteoform to the cell type and give us the single cell identity and eventually potentially phenotypes. So what we do is that it's quite like a similar workflow, but we did an augmentation of the platform to basically optimize the workflow, the liquid flow rate, and our visualization capabilities to verify single cells. And eventually, we are able to, see single cell proteoform detection by a immobilized single cell on a glass slide. So on top of the slide, you can see this video that shows a dynamic liquid bridge from the bottom of the slide that basically when we first park our liquid bridge next to the cell, we don't see any ion count or very low ion count from the background in this chronogram. And then once we move this liquid extraction bridge to the cell, which is highlighted here on top, you can see this sudden increase in the number of ions detected, and they're all proteoform ions. And after about 8 seconds, the signal detection wanes and goes back to the baseline. So this is what we call a cellogram, corresponds to the chronogram of detection of a single cell proteoform content. And then as you imagine that, if you are able to just put one single cell detection event to another without a space, then you can actually you just spend 8 seconds per cell, and you can analyze thousands of cells per day. So for now, with a random distribution of single cells on the glass slide, we are able to profile about 1 to 2,000 cells per day in the imaging-like mode, and every cell will give us about 20,000 ions. So I also wanted to mention that this platform has been significantly upgraded in Neil's lab with a lot of amazing development from our postdocs. Including VG and Claudia. So they are and graduate student Hannah. So they are able to really put the cells into regular patterns and really save time and eliminate the downtime between single cell detection to achieve thousands of ions, thousands of cells per day. So as I mentioned that every single cell detection has about 20,000 ions per cell. And if you sort of parse all these ions into different proteoforms, different isotopic distributions, you might imagine that we will have a hard time having enough copies of ions for a proteoform, right? And this is exactly what we encountered. And this is an example of a spectrum that we showed in the range of a NIH3T3 cell of 53.6 to in that range, 53.6 kilodalton. You will see these purple distributions corresponding to 31 single ions. And I'm using this kind of different level of blue colors to highlight different number of ions that you see corresponding to different peaks. And we know that there's a really abundant kind of proteoform marker of the cell type, which is vimentin, that's acetylated without the methionine initiator. So, and you can see some alignment between this group of cell ions with the theoretical isotopic distribution in dark. So how do we actually correlate these detection from these single ions to the assignment of the vimentin, identity? So we together with our informatics team and in Neil's lab, we developed this PAT score algorithm that basically evaluates the likelihood of a group of single ions to be assigned to a theoretical isotopic distribution. And this is by evaluating the combinatorial single ion alignment with the theoretical distribution of isotopes. So I'm not going to go through all the details of the equation, but on the right side, you can see kind of one example from a group of single ion that has one ion here that's aligned with its nearby theoretical isotope of a proteoform. And then we generate a probability score by evaluating the isotope intensity and the mass shift. And then all combined together with all the 31 ions, we can get a score for that proteoform assignment. And if you imagine that you have a amazing alignment of your ions with the distribution, you will get a score that is very close to 1. If you have like barely any alignment, then you will get a score of 0. And other assignment will kind of fall in between. And this will give us a kind of semi-quantitative approach to align a probability score to the likelihood or the intensity of a proteoform detection in a single cell. And we were adventurous enough, joined by a collaboration with Professor Jonathan Sweetler's group at University of Illinois at Urbana-Champaign. So we wanted to try a really complex sample that's from a actual animal with actual single cells that are live before we kill them. And these are the real, like biologically relevant cells that have different, cell types. So Jonathan's group are the expert in dissociating a brain tissue from a rat hippocampus to obtain live, like undisrupted neurons, astrocytes, and other cell types in the brain. And then we have a way to put them down onto a slide in different spatial locations and fix them, make them chemically compatible for proteoform detection. And then we apply single cell PIMS to analyze the proteoform content of each individual single cells. And so on top, this is an animation of how you scroll over different cells to get the proteoform content using the liquid extraction bridge. And at the bottom, it shows an actual video of the liquid bridge running over the two cells, one after another, without a carryover. And then this actually gives us the cellogram that shows two, proteoform detection event from the single cells. And this one corresponding to the first cell, and this one corresponds to the second cell. And with this kind of throughput, we are able to profile a total of 5,200 single cells from the rat brain over 5 days, and a total of 165 proteoforms are detected and assigned in all the single cells. And by a very sophisticated informatics approach in collaboration with our informatics group, we were able to correlate these proteoforms, calculate their scores, correlate them to different biological pathways with weight, and then eventually separate them into cell types. So we found a majority of the cells can be assigned to neurons, and others can be assigned to astrocytes and microglia. And we were able to
Apologies,
classify
Pei. We
more
have about
than
2 minutes.
Sure. I'm wrapping up here. So more than 90% of the single cells can be classified from this sample. Okay, so I'm going to skip some of these single cell work that talks about the assignment of the astrocyte and microglia. But I do actually want to mention that microglia has a very small population in our total, like 5,000 cells. It's less than 5%. So we really have an amazing dynamic range that can be able to like tease out just less than 5% of the cell type population from the brain. All right. So with that, I hope I was able to convince you how single molecule MS using Orbitrap systems is amazing for proteoform detection, not only in like a single component kind of protein sample with a complex proteoform landscape, but also it can be applied to spatial proteoform detection and the single cell proteoform detection with high throughput. And I wanted to talk about a little of the perspective work in my lab at UC Riverside. So we wanted to look into this kind of integrative approach to understand the biology of proteoforms, and this requires the development of probes that can profile proteoforms across different biological scales, from tissues to single cells to subcellular content. And we are planning to couple different, spatially resolved sampling probes using laser, liquid microextraction to single molecule mass spec. And this will eventually allow us to connect the proteoform biological roles with other proteins and other small molecules. And we are very excited to have our mass spec installed this week. We are currently still testing and calibrating the system, and very much looking forward to what it will allow us to do in the foreseen month. And the one work that I wanted to quickly go over is that the
Apologies,
lasers.
Pei. We're at 5 o'clock now. We need to
Okay.
we just need to stop.
Okay, sure. Sounds good. So, all right. I just want to quickly say thank you, acknowledgement to my young group and also my mentors, collaborators, funding resources, and I am ready to take questions and thank you so much for your attention.
Thank you for an excellent talk. That was a real tour de force. I love
Thank you.
spatial proteomics, so it's great to see. Uh,
Yes, and yeah, of course. I think I've also re read your recent papers on the spatial proteomics of different class of proteins from soluble to membrane proteins, and I think that's like really opening up the ability of our spatially sampling technologies.
what do you think is the biggest challenge to making this more available to more people, more labs?
So I think one of the challenges that we are developing new, spatially sampling ion sources, and these are highly custom systems, right? So I think really, we wanted to be able to work with, the instrument vendors to streamline, commercialize these, platforms, and also reduce the cost of a lot of these components if someone were to custom build these ion sources. And I actually mentioned this at like an earlier conference at UC Davis, like the International Mass Spec Imaging Conference, so that we actually can, make say like the nano-DESI platform with a relatively inexpensive kind of components and put together them within like just a few $1,000s. So this will basically like open up this like the kind of the usership, right? And also Julia's group has been like they published a few like tutorial and protocol papers to really like introduce the instrumentation to the community to make it more broadly applicable and adoptable in our community.
I agree. I think it's not a cost issue so much as a usability issue and giving people the right tools that they can just dive into experiments, not worry about it being delicate and difficult to use. I think that's the future of this.
Yeah, for sure.
So
And this is actually also like some directions that my group is very interested in pursuing in the future.
excellent. Okay, so we're on the hour, well, an hour since we started. So, and I don't see any questions in the chat. So, unless there was anything specifically more, we can end there. Thank you once again, Pei, for such an excellent talk. And so, hopefully, people will join us for the workshop next month and for the next Proteoform Thursday in March. So, that's all for now. Thank you.